Welcome back to Science with Sleepy, where curiosity drifts like starlight, and tonight's journey stretches across 2 full hours, because the universe of cellular programming never runs out of wonders. Imagine the lights dimmed low, a gentle hum of ambient sound behind my voice, and your favorite blanket pulled close, as we drift into one of the most fascinating questions in modern science. Picture this. You write a line of code on your computer, and the machine executes it perfectly, every single time. The code says, "Display this text," and the text appears. The code says, "Calculate this equation," and the answer appears. Computers are predictable, reliable, obedient machines that follow our instructions precisely. Now picture something entirely different. A living cell, floating in a drop of water. It's not silicon and metal. It's not programmed by humans. It's a tiny bag of molecules, constantly moving, constantly changing, constantly responding to its environment. It has its own needs, its own behaviors, its own will to survive. What if we could bridge these 2 worlds? What if we could program cells like we program computers? What if we could write code that tells living cells what to do, how to behave, when to produce certain molecules, and how to respond to their environment? What if cells could become programmable machines, executing our instructions as precisely as computers do? This is not science fiction. This is happening right now in laboratories around the world. Scientists are learning to program cells. They're creating biological circuits that work like computer circuits. They're writing genetic code that controls cell behavior. They're building systems where cells can count, remember, oscillate, and perform computations. Tonight, we're going to explore exactly that. We're going to wander slowly through how cells already work like tiny biological computers, how scientists are learning to reprogram them, and what might be possible if we could truly program life itself. We'll drift from the simple idea that cells process information, all the way to the complex systems where cells become factories, where bacteria produce medicines, where our own cells can be reprogrammed to fight disease. The story begins with a gentle question: what does it mean for a cell to be programmable? When you write computer code, you're giving instructions to a machine made of silicon and metal. When you program a cell, you're giving instructions to a machine made of molecules and proteins, a machine that's alive, that grows, that evolves. It's an interesting question because it sits at the intersection of 2 worlds. On one side, we have computer programming, where we write instructions in code, and machines execute them precisely. On the other side, we have biology, where living cells follow instructions written in DNA, instructions that evolved over billions of years. But these 2 worlds are not as different as they might seem. Both involve information processing. Both involve instructions being executed. Both involve systems that take inputs, process them, and produce outputs. The language is different, the materials are different, but the fundamental concepts are remarkably similar. As we drift through this long, gentle exploration, we will discover how cells already process information like computers do. We will talk about DNA, the biological programming language with just 4 letters. We will visit CRISPR, the revolutionary tool that lets us edit DNA like a word processor. We will explore synthetic biology, the field where biology meets engineering. And we will look at nanotechnology, where we program matter at the smallest scales. The study of cellular programming is one of the most active areas in modern science. Every year, new techniques appear, new capabilities are unlocked, and our understanding deepens. Yet the core question stays the same. How can we program life? What is actually happening when we give cells new instructions? This question touches the way our own bodies work, the nature of life itself, and what might be possible in the future. As we explore tonight, we are not just learning about cells. We are also learning about information, about control, about the relationship between design and evolution, and about the way simple rules can create very complex behavior. So settle in, let your breathing slow, and let your mind drift with me into the world of cellular programming. We are about to explore how cells process information, how scientists are learning to program them, and how this might change medicine, industry, and our understanding of life itself. The journey begins now, and it will take us from the simplest building blocks to the frontiers of what is possible, all in calm language that is easy to follow. Let's start with something simple, something fundamental. What is a cell? Close your eyes for a moment and imagine something incredibly small. So small that you could fit thousands of them on the tip of a pin. A cell is the smallest unit of life. It's a tiny, self-contained world, complete with its own machinery, its own instructions, its own purpose. Your body contains trillions of cells. Each one is like a tiny factory, constantly working, producing molecules, responding to signals, maintaining itself, and sometimes dividing to create new cells. If you could shrink yourself down and walk inside a cell, you would see a bustling city of activity. Molecules moving everywhere, proteins building structures, energy being produced and consumed, information flowing from place to place. But here's what makes cells remarkable from a programming perspective. Each cell follows instructions. Not instructions written by humans, but instructions written in DNA, a biological programming language that evolved over billions of years. Inside every cell, there's DNA. DNA is like a biological instruction manual, but it's written in a language with just 4 letters: A, T, G, and C. These letters stand for 4 molecules called nucleotides: adenine, thymine, guanine, and cytosine. The sequence of these letters encodes information, just like the sequence of ones and zeros encodes information in computer code. DNA contains genes, which are like programs that tell the cell what proteins to make. Proteins are the molecules that do most of the work in cells. They build structures, carry out chemical reactions, send signals, and respond to the environment. Think of proteins as the workers in the cellular factory, and genes as the instructions that tell those workers what to do. When you write computer code, you're giving instructions to a machine made of silicon and metal. The code says, add these numbers or display this text, and the computer executes those instructions precisely. When a cell reads its DNA, it's following instructions written in a biological language. The cell reads a gene, translates it into a protein, and that protein carries out a specific function. The language is different, the materials are different, but the concept is remarkably similar. Both involve instructions that get executed, that produce outputs, that control behavior. The difference is that computer code was designed by humans, while DNA evolved through natural selection. Computer code runs on machines we built, while DNA runs on molecular machinery that evolved over billions of years. But the fundamental idea is the same: instructions being read and executed, producing specific behaviors. This similarity has fascinated scientists for decades. In the 1960s, researchers began thinking about cells as information processing systems. They realized that cells take inputs, process information, and produce outputs, just like computers do. A cell might sense a chemical signal from its environment, process that information through complex biochemical pathways, and respond by producing a protein or changing its behavior. This realization led to a new way of thinking about biology. Instead of just studying what cells do, scientists began thinking about how cells process information. They began seeing cells as biological computers, following genetic programs that control their behavior. This was a profound shift in thinking. Biology had traditionally been a descriptive science, focused on understanding what exists in nature. But if cells process information like computers, maybe we could design biological systems like we design computer systems. Maybe we could write genetic code like we write computer code. This idea simmered for decades, slowly gaining traction as our understanding of genetics and molecular biology deepened. But it wasn't until around the year 2000 that it crystallized into a new field called synthetic biology. The term synthetic biology was popularized by researchers at MIT, Stanford, and UC Berkeley, who began treating biology as an engineering discipline. Instead of just studying biological systems, they began designing and building new ones. Instead of just observing what nature created, they began creating new biological parts, devices, and systems. Synthetic biology is the engineering of biological systems. It's about designing and building new biological parts, devices, and systems, or redesigning existing ones for useful purposes. It's like computer programming, but instead of writing code for silicon chips, you're writing code for living cells. The field has grown rapidly. According to research published in Nature Biotechnology, the number of synthetic biology papers published annually increased from about 100 in 2000 to over 2,000 by 2020. That's a 20-fold increase in just 2 decades. Companies in this space have raised billions of dollars in funding, and the global synthetic biology market is projected to reach over $30 billion by 2028. As of 2024, the synthetic biology industry is experiencing explosive growth. The market size was estimated at approximately $15 billion in 2024, with projections showing it could reach $50 billion or more by 2030. This growth is driven by advances in gene editing technologies, decreasing costs of DNA synthesis, and increasing applications across medicine, agriculture, and industrial biotechnology. Government investment has been substantial. The National Nanotechnology Initiative, established in 2000, has invested over $30 billion in nanotechnology research and development by 2024. This investment has supported thousands of research projects, leading to breakthroughs in materials, science, medicine, electronics, and environmental applications. The NNI has also launched the Nano 4 EARTH program, focusing specifically on nanotechnologies that address environmental challenges like energy storage, catalysis, and greenhouse gas capture. But what does it actually mean to engineer a biological system? What does it mean to program a cell? To understand this, we need to understand how cells already process information, how they already work like biological computers. To understand how we might program cells, we first need to understand how cells already process information. Inside every cell, there are networks of molecules that work together, like circuits. These biological circuits sense inputs, process signals, and produce outputs. Think about how a computer circuit works. You have inputs, like signals from a keyboard or mouse. These inputs flow through logic gates, which process the information. And then you get outputs, like text appearing on a screen, or a file being saved. The circuit takes information in, processes it according to rules, and produces a result. Biological circuits work similarly, but instead of silicon and metal, they're made of proteins and molecules. A cell might sense a chemical signal from outside. This signal binds to a receptor protein on the cell's surface, like a key fitting into a lock. The receptor changes shape, activating other proteins inside the cell. These proteins activate still more proteins, creating a cascade of activation. This cascade is like information flowing through a circuit. Eventually it leads to an output, like the cell producing a specific protein, or changing its behavior. Let's think about a concrete example. Imagine a bacterium floating in a solution. There's sugar in the solution, and the bacterium needs energy. The bacterium has a biological circuit that senses sugar. When sugar molecules bind to receptors on the cell surface, this triggers a cascade of protein activations inside the cell. The cascade eventually reaches the genes that code for enzymes that break down sugar. These genes get turned on, the enzymes get produced, and the bacterium starts metabolizing the sugar. This is information processing. The input is the presence of sugar. The processing is the cascade of protein activations. The output is the production of enzymes. It's a biological circuit, processing information just like a computer circuit does. Scientists have discovered that these biological circuits can be quite complex. Some circuits act like switches, turning genes on or off. Others act like timers, controlling when things happen. Still others act like logic gates, responding only when multiple conditions are met. For example, a cell might have a circuit that says, if there's sugar present, and if there's no oxygen, then produce this protein. This is like a simple computer program. It has conditions, it has logic, and it has an output. The cell processes multiple inputs, evaluates them according to logical rules, and produces a response. The difference is that biological circuits evolved over billions of years. They weren't designed by humans. They emerged through natural selection, through trial and error, through countless generations of life adapting to its environment. They're optimized for survival and reproduction, not for executing human-designed programs. But now, scientists are learning to design these circuits themselves. They're learning to program cells, to write genetic code that controls cell behavior. They're creating synthetic biological circuits that work like computer circuits, but made of molecules instead of silicon. The key to programming cells is understanding DNA. DNA is written in a language with just 4 letters: A, T, G, and C. These letters stand for 4 molecules called nucleotides: adenine, thymine, guanine, and cytosine. The sequence of these letters encodes information, just like the sequence of 1s and 0s encodes information in computer code. But here's what's remarkable. With just these 4 letters, nature has encoded all the information needed to build every living thing on Earth. Every plant, every animal, every bacterium, every human. All the diversity of life, all encoded in sequences of just 4 letters. When a cell needs to make a protein, it reads a gene from its DNA. The gene is like a recipe, written in this 4-letter language. It tells the cell which amino acids to string together to build the protein. There are 20 different amino acids, and proteins can be hundreds or thousands of amino acids long. The sequence of letters in the gene determines the sequence of amino acids in the protein, which determines the protein's shape and function. The cell has machinery that reads the DNA, translates it into RNA, and then uses the RNA to assemble the protein. This process happens constantly in every cell. Thousands of different proteins are being produced at any given moment, each one coded for by a specific gene. This process is called gene expression. When a gene is expressed, it means the cell is reading that gene and producing the protein it codes for. Cells can control which genes are expressed and when. They can turn genes on or off, speed up or slow down protein production, and coordinate the expression of many genes at once. This control is crucial. A cell doesn't need all its proteins all the time. It needs different proteins at different times, in different amounts, in response to different conditions. The cell regulates gene expression to produce exactly what it needs, when it needs it. To program a cell, scientists need to be able to control gene expression. They need to be able to turn genes on or off on command. They need to be able to make cells produce specific proteins at specific times. They need to be able to design new genes that do new things. This is the essence of cellular programming. You write genetic code that controls when genes are expressed, which proteins are produced, and how cells behave. It's like writing a computer program, but instead of controlling pixels on a screen or data in memory, you're controlling the molecular machinery of life. This is where genetic engineering comes in. Genetic engineering is the process of modifying an organism's DNA. Scientists can add new genes, remove existing genes, or modify genes to change how they work. They can insert DNA from one organism into another. They can design entirely new DNA sequences that have never existed in nature. The tools for doing this have been developing for decades. In 1970, scientists discovered restriction enzymes, proteins that can cut DNA at specific sequences. These enzymes act like molecular scissors, recognizing specific DNA sequences and cutting them precisely. In 1972, Paul Berg at Stanford created the first recombinant DNA molecule by combining DNA from different sources, work that earned him the Nobel Prize in Chemistry in 1980. In 1973, Stanley Cohen at Stanford and Herbert Boyer at UCSF developed techniques to insert foreign DNA into bacteria, creating the first genetically engineered organisms. This work laid the foundation for the entire biotechnology industry. In the 1980s, Kary Mullis invented the polymerase chain reaction, or PCR, a technique that allows scientists to copy specific genes millions of times in just hours. This revolutionized genetic research and diagnostics. PCR is now used in everything from paternity testing to COVID-19 detection. In the 1990s, the Human Genome Project began sequencing entire genomes, reading all the DNA in an organism. Completed in 2003, this project sequenced all 3 billion base pairs of human DNA, providing a complete map of human genetic information. Today, sequencing a human genome costs less than $1,000 and takes just a few days, compared to the $3 billion and 13 years it took for the first human genome. But the real breakthrough came in 2012 with a technology called CRISPR-Cas9, developed by Jennifer Doudna and Emmanuelle Charpentier, who won the Nobel Prize in Chemistry in 2020 for this work. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a system that bacteria use to defend themselves against viruses, but scientists have adapted it to edit DNA with incredible precision. CRISPR works like a pair of molecular scissors guided by a GPS system. Scientists can program it to cut DNA at any location they choose by designing a guide RNA molecule that matches the target DNA sequence. They can then insert new DNA sequences, delete unwanted sequences, or modify existing ones. It's like having a word processor for DNA, where you can find any word, delete it, replace it, or add new text. This technology has made genetic engineering much easier, faster, and more precise. Before CRISPR, editing a specific gene could take months or years. Now it can take days. Scientists can now edit DNA in ways that were impossible just a few years ago. They can program cells to do new things, to produce new molecules, to respond to new signals. As of 2024, CRISPR technology has moved beyond research into clinical applications. The first CRISPR-based therapy, Casgevy, was approved by the FDA in December 2023 for treating sickle cell disease and beta thalassemia. This marked a historic milestone, the first approved therapy using CRISPR gene editing. The therapy involves extracting a patient's stem cells, editing them with CRISPR to correct the genetic mutation, and reinfusing them. Clinical trials showed remarkable results, with many patients achieving complete remission from these previously incurable diseases. In 2024, researchers successfully used CRISPR delivered via lipid nanoparticles to treat a child with a rare genetic disorder, correcting a faulty liver enzyme. This represented personalized gene therapy made possible by programmable nanotechnology, demonstrating that CRISPR could be delivered directly into the body without needing to extract and modify cells in the laboratory. Beyond therapeutic applications, CRISPR is being used for molecular diagnostics and pathogen detection. CRISPR-based diagnostic tests can detect viruses, bacteria, and genetic mutations with high accuracy and speed. These tests are being developed for rapid disease diagnosis, food safety testing, and environmental monitoring. The CRISPR market itself is growing rapidly. As of 2024, the global CRISPR technology market was valued at over $5 billion and is projected to reach $15 billion by 2030. This growth reflects both the expanding therapeutic applications and the increasing use of CRISPR in research, agriculture, and industrial biotechnology. So what does it actually mean to program a cell? Let's think about a real example that's been happening for decades, an example that changed medicine forever. In 1978, scientists at Genentech successfully inserted the human insulin gene into the bacterium E. coli. This was one of the first major achievements in genetic engineering, and it demonstrated that we could program cells to produce molecules that they would never naturally produce. Here's how it works, step by step. Scientists take the gene that codes for human insulin. This gene contains the instructions for building the insulin protein, a hormone that regulates blood sugar. The gene is just a sequence of A's, T's, G's, and C's, but it encodes all the information needed to build insulin. Scientists insert this gene into a bacterium's DNA, using special enzymes that can cut and paste DNA. They also add a promoter, a piece of DNA, that controls when the gene is turned on. Think of the promoter as a switch. When the bacterium grows in certain conditions, the promoter activates, and the bacterium starts reading the insulin gene and producing insulin. The bacterium doesn't know it's making insulin. It doesn't understand what insulin is or why it's important, it's just following its DNA instructions. It reads the gene, translates it into RNA, uses the RNA to assemble the insulin protein, and releases it into its environment. From the bacterium's perspective, it's just doing what its DNA tells it to do. But from our perspective, we've programmed the bacterium to be a tiny insulin factory. We've given it instructions, and it's executing them. We've taken a living cell and programmed it to produce a specific molecule on demand. This is cellular programming in its simplest form. You insert a gene, you control when it's expressed, and the cell produces the protein. It's like writing a simple computer program. If certain conditions are met, then produce this output. This technology was approved by the FDA in 1982, and today, most insulin used by people with diabetes is produced this way. Before this, insulin was extracted from the pancreases of pigs and cows, which was expensive, limited in supply, and sometimes caused allergic reactions. The first genetically engineered insulin, called Humulin, was produced by Eli Lilly, using bacteria programmed with the human insulin gene. It's safer and more reliable than extracting insulin from animal pancreases. Many other medicines are now produced this way too. According to the Biotechnology Innovation Organization, over 200 biotech drugs and vaccines are currently produced using programmed cells. These include human growth hormone, erythropoietin for treating anemia, clotting factors for hemophilia, and many vaccines. Bacteria and yeast cells are programmed to produce proteins used as drugs, hormones, vaccines and enzymes. These cells are like biological factories, churning out molecules that would be difficult or expensive to make any other way. The global market for recombinant proteins produced by programmed cells is substantial. As of 2024, the biopharmaceutical market, which includes proteins produced by programmed cells, was valued at over $400 billion globally. This includes monoclonal antibodies, vaccines, hormones, enzymes, and other therapeutic proteins. The market is growing at approximately 8 to 10% annually, driven by increasing demand for biologics and advances in manufacturing technology. The insulin market alone, which is almost entirely produced using programmed cells, was valued at approximately $25 billion in 2024. This includes both human insulin and insulin analogs, all produced using genetic engineering techniques. The market continues to grow as diabetes rates increase worldwide, demonstrating the ongoing importance of cellular programming for medicine production. Beyond pharmaceuticals, programmed cells are used to produce industrial enzymes, food ingredients, biofuels, and specialty chemicals. The industrial biotechnology market, which relies heavily on programmed cells, was valued at over $200 billion in 2024 and is projected to reach $400 billion by 2030. This includes everything from enzymes for laundry detergents to bio-based plastics to renewable fuels. But scientists want to go further. They want to program cells to do more complex things. They want cells that can sense multiple inputs, process information and make decisions. They want cells that can work together, that can form circuits, that can perform computations. This is where synthetic biology gets really interesting. Scientists are designing biological circuits that work like computer circuits. They're creating genetic programs that control cell behavior. They're building systems where cells can count, remember, oscillate, and respond to complex patterns. For example, in 2000, Michael Elowitz and Stanislas Leibler at Princeton created the repressolator, the first synthetic biological oscillator. This was a landmark achievement, proving that cells could be programmed to perform time-based functions. Here's how the repressolator works. They programmed E. coli bacteria with 3 genes that repress each other in a cycle. Gene A represses gene B. Gene B represses gene C. And gene C represses gene A. This creates a cycle, a feedback loop that oscillates. When gene A is active, it represses gene B, which allows gene C to become active, which represses gene A, which allows gene B to become active, and so on. The cycle repeats, creating a biological clock that produces proteins in regular waves, like a metronome. This might seem simple, but it was revolutionary. It showed that we could program cells to perform time-based functions, to create rhythms, to oscillate. It proved that biological circuits could work like electronic circuits, with feedback loops and oscillations. The repressolator became a foundational example in synthetic biology, showing what was possible. In 2005, researchers at MIT created bacteria that could detect light and move toward it, by programming them with light-sensitive proteins. They inserted genes from photosynthetic bacteria into E. coli, giving the bacteria the ability to sense light. When light hit the cells, it triggered a cascade of protein activations that caused the bacteria to move toward the light source. This was like programming cells to be phototactic, to respond to light, opening up possibilities for light-controlled biological systems. In 2009, scientists at Boston University created a genetic circuit that could count how many times a cell had divided. They programmed cells with a memory system that recorded each division. The circuit used a series of genetic switches that flipped sequentially with each division, creating a binary counter. After one division, one switch flipped. After two divisions, two switches flipped. The cell could remember its division history, essentially counting its own divisions. This showed that cells could be programmed to have memory, to store information, to count events. In 2013, scientists at Stanford created cells that could perform simple computations, storing and processing information like a biological computer. They programmed cells with genetic circuits that could perform logic operations like AND, OR, and NOT. They could combine these operations to create more complex computations. For example, a cell could be programmed to produce a protein only if two conditions were met, implementing an AND gate. Or it could produce a protein if either of two conditions was met, implementing an OR gate. This showed that cells could be programmed to perform computations, to process information, to make decisions based on multiple inputs. In 2016, researchers used MIT's Cello programming language to design 60 different genetic circuits, and 45 of them worked as predicted when tested in living cells. This was remarkable. It showed that programming cells was becoming more reliable, more predictable. Scientists could write genetic code using a programming language, compile it into DNA sequences, insert it into cells, and have it work as expected. The success rate of 75% might seem low compared to computer programming, but for biological systems, it was impressive. It showed that cellular programming was moving from art to engineering, from trial and error to systematic design. More recently, in December 2024, researchers at Pohang University of Science and Technology in South Korea developed the Synthetic Translational Coupling Element, or SYN-TCE. This technology improves the precision of synthetic genetic circuits by accurately transmitting input signals to downstream genes, enabling simultaneous control of multiple inputs and outputs within a single RNA molecule. This advancement allows for more complex programming of cells, with applications in customized cell therapeutics and biofuel production. These are real experiments, published in scientific journals, that prove programming cells is possible. But they show the potential. If we can program cells to do simple things, maybe we can program them to do complex things. Maybe we can program them to diagnose diseases, to produce medicines on demand, to clean up pollution, to build materials. And this is already happening. In 2020, Moderna and Pfizer-BioNTech used lipid nanoparticles, tiny programmed structures, to deliver mRNA instructions to cells. This was a form of cellular programming that helped create vaccines in record time, demonstrating the power of programmable biology. As of 2024, mRNA vaccine technology has expanded far beyond COVID-19. Over 200 mRNA-based therapies are now in clinical development, targeting everything from cancer to infectious diseases to rare genetic disorders. Moderna alone has over 40 mRNA programs in development, including personalized cancer vaccines, vaccines for respiratory viruses, and treatments for rare diseases. The mRNA therapeutics market was valued at approximately $50 billion in 2024, largely driven by COVID-19 vaccines, but with significant growth expected from new applications. Cancer vaccines are a major focus, with personalized mRNA vaccines that are tailored to each patient's specific tumor mutations, showing promising results in clinical trials. These vaccines program immune cells to recognize and attack cancer cells, potentially revolutionizing cancer treatment. The technology is also being applied to rare diseases. mRNA therapies can program cells to produce missing proteins, potentially treating genetic disorders like cystic fibrosis, muscular dystrophy, and metabolic diseases. Early clinical trials have shown promise. with some patients experiencing significant improvements in symptoms. The success of mRNA vaccines has also accelerated investment in lipid nanoparticle technology. Companies are developing next-generation lipid nanoparticles with improved stability, better targeting, and reduced side effects. The lipid nanoparticle market itself is projected to grow from $1.5 billion in 2024 to over $5 billion by 2030, driven by the expanding applications of mRNA therapeutics. Here's how it worked. Scientists designed mRNA molecules that coded for the COVID-19 spike protein. This mRNA was like a program, instructions for cells to produce a specific protein. But mRNA is fragile. It would be destroyed if injected directly into the body. So scientists wrapped it in lipid nanoparticles, tiny spheres about 100 nanometers across, made of fat molecules. These nanoparticles were carefully engineered. They had to protect the mRNA, deliver it to cells, and release it at the right time. The nanoparticles were injected into muscle tissue, where they were taken up by cells. Once inside a cell, the nanoparticle released the mRNA. The cell's machinery read the mRNA instructions and produced the spike protein. The immune system saw this foreign protein and learned to recognize it, creating immunity. This was cellular programming in action. Scientists wrote genetic code, packaged it in nanoparticles, delivered it to cells, and the cells executed the program, producing the desired protein. It was like uploading software to biological hardware, programming cells to produce a vaccine component. The speed of this development was remarkable. Traditional vaccines take years to develop, but mRNA vaccines were designed in days, tested in months, and deployed within a year. This was possible because the platform was programmable. Once scientists understood how to program cells with mRNA, they could quickly design new vaccines for different viruses. The same platform that worked for COVID-19 could work for influenza, for cancer, for other diseases. It was like having a programming language for vaccines, where you could write new code for new diseases. This showed the power of programmable biology. When you can program cells, you can respond quickly to new challenges. You can design new treatments rapidly. You can adapt biological systems to new purposes. The COVID-19 vaccines were just the beginning. Scientists are now developing mRNA vaccines for cancer, for personalized medicine, for diseases we haven't even imagined yet. Let's think about what programming cells might look like in the future, and what's already happening today. Scientists are developing programming languages for biology, tools that let us write genetic code like we write computer code. For example, researchers at MIT created a programming language called cello. The name comes from the idea of orchestrating biological functions, like a conductor orchestrating a cello. Cello lets scientists write genetic programs, using code similar to computer programming languages. You might write something like, if the cell detects a cancer marker, then produce a protein that kills cancer cells. If the cell detects inflammation, then produce an anti-inflammatory molecule. If the cell detects low oxygen, then produce a protein that stimulates blood vessel growth. This is like writing a program with if-then statements, but the program runs inside your body, inside your cells. The cells become tiny computers, constantly monitoring their environment and responding appropriately. You write the code, compile it into DNA sequences, insert it into cells, and the cells execute the program. The cello language works by translating high-level genetic programs into DNA sequences. You write your program using logical operators like AND, OR, and NOT. Cello then designs genetic circuits that implement these operations, selecting appropriate promoters, repressors, and other genetic parts. It's like a compiler for biology, translating human-readable code into genetic sequences. In 2016, researchers used Cello to design 60 different genetic circuits, and 45 of them worked as predicted when tested in living cells. This 75% success rate showed that programming cells was becoming more reliable and predictable. Scientists could write code, compile it, and have it work in living cells. It was a major step toward making cellular programming as routine as computer programming. But Cello is just the beginning. Other programming languages for biology are being developed. Some focus on specific applications, like programming immune cells or programming bacteria for industrial use. Others aim to be more general purpose, allowing scientists to program any type of cell for any purpose. The goal is to make programming cells as easy as programming computers. Imagine a future where you can download genetic code from a repository, modify it for your needs, and upload it into cells, where biological programs are shared, improved and combined, just like software today, where you can program cells using familiar programming concepts, without needing deep expertise in molecular biology. This future is coming. Programming languages for biology are getting better, the tools are improving, the success rates are increasing. We're moving toward a world where programming cells is as accessible as programming computers. Scientists have also developed synthetic receptors that can sense specific molecules and trigger cellular responses. One example is the synthetic Notch receptor, or SynNotch, developed by researchers at UCSF. These receptors can be programmed to detect specific molecules, like cancer markers, and trigger the cell to produce therapeutic proteins, or change its behavior. In 2016, researchers used SynNotch receptors to program cells to self-assemble into specific multicellular structures, essentially programming cells to build tissues with desired architectures. This technology has been used to engineer cells that can detect and kill cancer cells more effectively. In November 2024, researchers published work on synthetic frequency-controlled gene circuits that can process information through frequency-modulated signals. These circuits convert frequency-modulated signals into amplitude-based responses, expanding the controllable gene expression space and allowing for more sophisticated cellular behaviors. This is like programming cells to respond to different patterns of signals, not just single signals, making cellular programming more like complex computer programming. There's also DNA nanotechnology, where scientists program DNA molecules themselves to fold into specific shapes and structures. This is programming at the molecular level, creating nanoscale machines from DNA. In 2006, Paul Rothemund at Caltech developed DNA origami, a technique where long strands of DNA are programmed to fold into precise nanoscale shapes. The name comes from the Japanese art of paper folding, but instead of paper, scientists fold DNA. They use a long single strand of DNA, called a scaffold, and program it with hundreds of short DNA strands, called staples. Each staple binds to specific parts of the scaffold, forcing it to fold into a desired shape. Rothman demonstrated this by creating DNA shapes like squares, triangles, stars, and even smiley faces, all just nanometers across. These weren't just artistic demonstrations, they showed that DNA could be programmed to form precise structures, opening up possibilities for nanoscale engineering. Since then, scientists have created increasingly complex DNA structures. They've built DNA boxes with lids that can open and close. They've created DNA robots that can walk along tracks, carrying cargo from one location to another. They've built DNA computers that can perform simple computations, processing information encoded in DNA sequences. These aren't cells, but they're biological molecules programmed to perform specific functions. They're nanoscale machines made of DNA, executing programs written in genetic code. In 2024, researchers published work on DNA origami-engineered nanomaterials for drug delivery and biosensing, showing how nanotechnology and synthetic biology are converging. DNA origami has practical applications. Scientists have created DNA boxes that can carry drug molecules and release them when they encounter specific conditions, like the presence of cancer cells. They've built DNA sensors that can detect specific molecules and produce a signal. They've designed DNA structures that can organize other molecules, creating nanoscale assembly lines. The precision of DNA origami is remarkable. Scientists can design structures with nanometer accuracy, positioning molecules exactly where they want them. This level of control opens up possibilities for creating new materials, new sensors, new drug delivery systems, all programmed at the molecular level. Even more recently, researchers have developed RNA origami, where RNA molecules are folded into specific nanoscale shapes within living cells. Developed by researchers from Aarhus University and Caltech, RNA origami allows for the creation of large RNA nanostructures that can serve as scaffolds for organizing RNA-based functionalities. This technique has potential applications in nanomedicine, synthetic biology, and even constructing artificial cytoskeletons for synthetic cells. Unlike DNA origami, RNA origami works inside living cells, making it potentially more useful for programming cellular functions. Scientists are also exploring DNA as a data storage medium, programming biological molecules to store digital information. This might seem like science fiction, but it's real, and it has remarkable potential. In 2021, researchers at Microsoft and the University of Washington successfully stored and retrieved 1 gigabyte of data in DNA. They encoded digital files into DNA sequences, synthesized the DNA, stored it, and later read it back, recovering the original files with perfect accuracy. DNA has incredible advantages as a storage medium. It can store information at a density millions of times greater than traditional hard drives. A single gram of DNA could theoretically store 215 petabytes of data, equivalent to about 215 million gigabytes. To put that in perspective, all the data in the world could fit in a few kilograms of DNA. DNA is also incredibly stable. Under the right conditions, DNA can last for thousands of years. Scientists have recovered readable DNA from fossils that are tens of thousands of years old. Compare that to hard drives, which degrade over decades, or flash memory, which can lose data over years. DNA could preserve information for millennia. DNA storage also requires no energy to maintain. Once the data is encoded in DNA, it just sits there, stable, requiring no power, no maintenance, no updates. It's like writing information in stone, but at the molecular level. The process works like this. Digital data, which is stored as ones and zeros, is encoded into DNA sequences, which use A, T, G, and C. Scientists have developed encoding schemes that translate binary data into genetic code. For example, they might encode 0 zeros as A, 01 as T, 10 as G, and 11 as C. The DNA is synthesized, stored, and later sequenced to read the data back. Currently, DNA storage is slow and expensive. Writing data to DNA takes hours or days, and reading it back takes similar time. The cost is also high, though it's decreasing rapidly. Synthesizing DNA costs thousands of dollars per megabyte, though researchers are working to reduce this cost by orders of magnitude. But for long-term archival storage, where speed isn't critical, DNA could be ideal. Imagine storing all of humanity's knowledge in a few vials of DNA, preserving it for future generations. Companies like Microsoft and Twist Bioscience are actively developing DNA storage systems. Microsoft has demonstrated storing and retrieving data from DNA, and Twist Bioscience is working on making DNA synthesis cheaper and faster. The goal is to make DNA storage practical for long-term data archiving, where the incredible density and stability of DNA make it superior to traditional storage media. While still experimental, DNA storage shows how biological molecules can be programmed to store and process information, just like computers do. It's another example of how biology and computing are converging, how we're learning to use biological systems for computational purposes. In the future, we might store our most important data in DNA, preserving it for thousands of years, ensuring that our knowledge survives, even if our technology doesn't. Nanotechnology takes programming even smaller, working at the scale of individual molecules. A nanometer is 1/1,000,000th of a meter, about the size of a few atoms. To put this in perspective, a human hair is about 100,000 nanometers wide. A red blood cell is about 7,000 nanometers across. A virus is typically 20 to 300 nanometers. At the nanoscale, we're working with individual molecules, programming matter at its most fundamental level. At this scale, scientists can program individual molecules to perform specific tasks. In 2024, researchers developed nanorobots equipped with artificial intelligence that can navigate complex biological environments. These nanorobots use reinforcement learning algorithms to analyze biomarker concentration gradients, allowing them to detect and target cancer cells with precision. They're essentially programmable molecular machines, smaller than cells, that can perform medical tasks. These AI-enhanced nanorobots represent a convergence of three cutting-edge technologies: nanotechnology, artificial intelligence, and synthetic biology. The nanorobots are made of nanomaterials, programmed with AI algorithms, and designed to interact with biological systems. They can navigate through the bloodstream, detect disease markers, and deliver therapeutic payloads precisely where needed. The development of these nanorobots required solving numerous challenges. They had to be small enough to navigate through blood vessels, stable enough to survive in the body, and smart enough to make decisions based on their environment. The AI component allows them to learn from their experiences, improving their performance over time. This is like having microscopic robots that can think, learn, and adapt, all while operating inside the human body. Current research is exploring nanorobots for various medical applications. Some are designed to deliver drugs directly to tumors, minimizing side effects. Others are designed to remove plaque from arteries, potentially preventing heart attacks. Still others are designed to repair damaged tissues at the cellular level. The possibilities are vast, and we're just beginning to explore them. Nanotechnology also enables better ways to program cells. Lipid nanoparticles, tiny spheres about 100 nanometers across, can carry genetic instructions into cells. The COVID-19 mRNA vaccines used this technology. The lipid nanoparticles protect the mRNA as it travels through the body, then fuse with cell membranes to deliver their genetic payload. Once inside, the cell reads the mRNA instructions and produces the spike protein. This is a form of temporary cellular programming, where cells are given new instructions that they execute for a short time before returning to normal. The lipid nanoparticles used in mRNA vaccines are carefully engineered. They consist of 4 types of lipids. Ionizable lipids that help the nanoparticles enter cells, helper lipids that stabilize the structure, cholesterol that provides fluidity, and PEG lipids that prevent the nanoparticles from being cleared by the immune system too quickly. The exact composition affects how well the mRNA is delivered, and how long it lasts in the body. This level of molecular engineering shows how nanotechnology allows us to program materials at the smallest scales to perform specific biological functions. Scientists are also developing single-cell nanoencapsulation, where individual living cells are wrapped in nanometer-thick shells. This technique provides isolation, protection, and functionalization of cells. By controlling the cellular microenvironment at the nanoscale, researchers can design engineered cellular systems with tailored properties. The process involves coating individual cells with thin layers of nanomaterials, creating protective shells that are just nanometers thick. These shells can be made from various materials, including polymers, lipids, or inorganic compounds. The shells protect the cells from immune attack, from harsh environments, or from other cells, while still allowing nutrients and signals to pass through. This has applications in cell therapy, where you want to protect transplanted cells from the immune system. Instead of suppressing the entire immune system, you can protect just the cells you're transplanting. The shells can also be functionalized, meaning they can be programmed to respond to specific conditions, to release drugs, or to interact with the environment in controlled ways. In regenerative medicine, nanoencapsulation can protect stem cells, as they differentiate into specific cell types. The shells can provide signals that guide differentiation, or they can protect the cells until they're ready to integrate into tissues. This could improve the success rate of stem cell therapies, which currently face challenges with cell survival and integration. In probiotics, nanoencapsulation can protect beneficial bacteria as they pass through the stomach, ensuring they reach the intestines where they're needed. The shells can also be programmed to release the bacteria at specific locations, or in response to specific conditions, improving the effectiveness of probiotic treatments. The technique is also being explored for agriculture, where nanoencapsulated cells could be used to deliver nutrients or protect plants from pathogens. By controlling how cells interact with their environment at the nanoscale, researchers can create more effective and targeted biological systems. Scientists are also creating nanocomposites, materials that combine nanoparticles with other substances to create new properties. For example, researchers have developed nanocellulose aerogels integrated with molybdenum disulfide for flame retardancy. These materials are programmed at the molecular level to have specific properties, like being lightweight yet strong, or being able to absorb water from air without external energy. In 2024, engineers at Penn Engineering discovered materials that can passively harvest water from air, using nanoscale structures that combine water-attracting and water-repelling components. These materials can continuously capture and release moisture without external energy, potentially providing water in arid regions. The nanocomposites industry is growing rapidly. According to market research, the global nanocomposites market is projected to grow from $9.15 billion in 2024 to $24.66 billion by 2032, representing a compound annual growth rate of 13.20%. These materials are increasingly used in automotive and aerospace industries, where their superior mechanical and optical properties make vehicles lighter, stronger, and more fuel efficient. In 2025, researchers at MIT developed a faster and more efficient method to manufacture nanoparticles that deliver cancer drugs directly to tumors. This new device can produce about 50 doses of nanoparticles in just a few minutes, meeting industry safety standards. This shows how nanotechnology manufacturing is becoming more practical and scalable, moving from laboratory curiosities to real medical applications. Or imagine programming bacteria to clean up oil spills. This is one of the most promising applications of cellular programming for environmental remediation. Oil spills are devastating to ecosystems. When oil leaks into the ocean, it forms slicks that kill marine life, contaminate beaches, and persist for years. Traditional cleanup methods involve skimming oil from the surface, using dispersants, or burning it, but these are often incomplete and can cause additional environmental damage. But what if we could program bacteria to eat the oil? What if we could release programmed bacteria that seek out oil, break it down into harmless components, and then die off when the job is done? Scientists are already working on this. Researchers have engineered bacteria that can break down oil by programming them with genes that produce enzymes called alkane hydroxylases. These enzymes can degrade hydrocarbons found in oil, breaking them down into simpler molecules that bacteria can use as food. The bacteria essentially eat the oil, converting it into harmless byproducts. In laboratory tests, programmed bacteria have successfully broken down crude oil components. They can degrade various types of hydrocarbons, from simple alkanes to complex aromatic compounds. The bacteria multiply as they eat, creating more oil-eating bacteria, accelerating the cleanup process. But the programming can be more sophisticated. Scientists are developing bacteria with genetic circuits that say, if you detect oil, then produce enzymes that break down oil. If you run out of nutrients, then stop and wait. If you detect other bacteria doing the same job, then coordinate with them. This creates intelligent cleanup systems that respond to conditions, that work together, that optimize their behavior. The bacteria would become a distributed computing system, working together to solve a problem. Each bacterium would be like a tiny processor, executing its program, communicating with others through chemical signals, and contributing to the overall task. Millions of bacteria, each running the same program, could clean up massive oil spills. However, using programmed bacteria in real oil spills is still being researched due to safety and regulatory concerns. What if the bacteria escape? What if they interact with natural ecosystems in unexpected ways? What if they evolve in ways we didn't anticipate? These are serious questions that need to be answered before widespread deployment. Scientists are addressing these concerns by designing safety features into the programmed bacteria. They're creating bacteria that require specific nutrients that don't exist in natural environments, so they'll die if they escape. They're programming bacteria with kill switches that can be activated remotely. They're designing bacteria that can't reproduce, so they can't establish themselves in the environment. Scientists have also created bacteria that can detect and break down other pollutants, bacteria that can remove arsenic from water, making it safe to drink, bacteria that can break down pesticides, cleaning contaminated soil, bacteria that can remove heavy metals, detoxifying polluted sites. In 2016, Japanese researchers discovered bacteria that had naturally evolved to eat plastic. These bacteria produce enzymes that can break down PET plastic, the kind used in water bottles. Scientists are now trying to program similar capabilities into other bacteria, creating plastic-eating bacteria that could help clean up the massive amounts of plastic waste in our oceans. The possibilities are vast. Programmed bacteria could clean up contaminated sites, remove pollutants from water, break down plastic waste, restore damaged ecosystems. They could work continuously, multiplying as they work, adapting to conditions, cleaning up pollution that would otherwise persist for decades or centuries. This is the future of environmental remediation, not just cleaning up pollution, but programming life to clean it up for us. using biological systems that work with nature, not against it. Or imagine programming cells to build materials. This is already happening, and it's transforming how we create materials. Instead of manufacturing materials in factories using heat, pressure, and chemicals, we can program cells to grow them, using biology's own manufacturing processes. Companies like Bolt Threads and AM Silk are already programming bacteria to produce spider silk proteins, which are incredibly strong. Spider silk is one of nature's most remarkable materials. Pound for pound, it's stronger than steel, more flexible than rubber, and lighter than most synthetic materials. But spiders are difficult to farm, and they produce only tiny amounts of silk, so scientists program bacteria to produce spider silk proteins instead. The process works like this. Scientists identify the genes that code for spider silk proteins. They insert these genes into bacteria, programming the bacteria to produce the proteins. The bacteria grow in fermentation tanks, producing the silk proteins as they multiply. Scientists then harvest the proteins and spin them into fibers, creating materials that have the remarkable properties of spider silk. These materials are being used in everything from medical sutures to high-performance textiles. Medical sutures made from spider silk proteins are stronger than traditional sutures, and are naturally biodegradable, so they dissolve as wounds heal. High-performance textiles made from spider silk proteins are being developed for applications in aerospace, defense, and sports equipment, where strength and lightness are critical. But spider silk is just the beginning. Scientists are programming cells to produce other remarkable materials. Some are programming bacteria to produce materials inspired by abalone shells, which are incredibly strong due to their layered structure. Others are programming cells to produce materials inspired by gecko feet, which can stick to surfaces without adhesives. Still others are programming cells to produce materials that can self-heal, repairing damage automatically. The advantages of biological material production are significant. It's more sustainable than traditional manufacturing, using less energy and producing less waste. It can create materials with properties that are difficult or impossible to achieve through traditional methods. And it can produce materials at ambient temperatures and pressures, reducing the environmental impact of manufacturing. The bio-based materials market is growing rapidly. As of 2024, the market for bio-based materials was valued at over $50 billion, and is projected to reach $100 billion by 2030. This includes everything from bio-based plastics, to bio-based textiles, to bio-based construction materials. Programmed cells are playing an increasingly important role in this market, as scientists learn to program cells to produce more and more types of materials. Scientists are already working on all of these ideas. Companies like Amyris are programming yeast cells to produce biofuels and renewable chemicals. Amyris has developed yeast strains that can convert sugar into farnesene, a molecule that can be used as a biofuel or converted into various chemicals. The company has demonstrated that programmed cells can produce these molecules at commercial scale, showing that cellular programming isn't just a laboratory curiosity, but a viable industrial technology. Companies like Novozymes program bacteria to produce enzymes for industrial processes. Novozymes is one of the world's largest producers of industrial enzymes, producing enzymes for everything from laundry detergents, to food processing, to biofuel production. These enzymes are all produced using programmed cells, demonstrating the commercial viability of cellular programming for industrial applications. Companies like Ginkgo Bioworks use automated systems to program cells for various applications, from producing fragrances to manufacturing pharmaceuticals. Ginkgo Bioworks has built automated facilities that can program cells at scale, using robotics and AI to design, build, and test genetic programs. The company has programmed cells to produce everything from fragrances to food ingredients to pharmaceutical intermediates, showing the versatility of cellular programming. Twist Bioscience synthesizes DNA sequences on demand, providing the raw materials for genetic programming. The company can synthesize DNA sequences thousands of base pairs long, providing researchers and companies with the genetic code they need to program cells. As DNA synthesis costs decrease, it's becoming easier and cheaper to program cells, democratizing access to cellular programming technology. The synthetic biology industry is maturing rapidly. What started as academic research is now becoming a commercial industry, with companies raising billions of dollars in funding and generating significant revenue. The industry is creating new products, new processes, and new possibilities, all based on the ability to program cells. But the industry also faces challenges. Scaling up from laboratory to commercial production is difficult. Programmed cells don't always behave as expected at scale. Manufacturing processes need to be optimized. Regulatory approval can be slow and expensive, but despite these challenges, the industry is growing, and programmed cells are becoming an increasingly important part of our economy. Researchers are programming immune cells to fight cancer more effectively through CAR T-cell therapy, which was approved by the FDA in 2017. Scientists have created bacteria that can detect toxins like arsenic in water and produce a visible signal, essentially creating living biosensors. In 2024, researchers developed carbon nanotube-based sensors capable of detecting plant stress signals, offering early warning systems for farmers. These sensors can identify when plants experience heat, light stress, or pathogen attacks, showing how nanotechnology can help monitor biological systems. Scientists have also developed ways to reprogram cells in vivo, meaning inside the living body, without needing to extract and modify cells in the laboratory. This is a major advance, because it eliminates the need for complex cell extraction, modification, and reinfusion procedures. By delivering modified mRNA into living organisms using nanoparticles, researchers can temporarily reprogram cells. The mRNA carries instructions for producing specific proteins, and when cells take up the mRNA, they produce those proteins. The reprogramming is temporary because mRNA degrades over time, but for many applications, temporary reprogramming is sufficient or even desirable. For example, scientists have reprogrammed circulating T cells in vivo to express chimeric antigen receptors, offering a potential strategy for personalized cancer immunotherapy that doesn't require the complex process of extracting, modifying, and reinfusing cells. Instead of removing T cells, modifying them in the laboratory, and reinfusing them, scientists can inject nanoparticles carrying mRNA that programs T cells directly in the body. This simplifies the treatment process and could make CAR T therapy more accessible. In vivo reprogramming is also being explored for other applications. Scientists are developing ways to reprogram liver cells to produce missing enzymes, potentially treating genetic disorders. They're exploring ways to reprogram muscle cells to produce therapeutic proteins. They're investigating ways to reprogram immune cells to fight infections or autoimmune diseases. The advantage of in vivo reprogramming is that it's less invasive and more accessible than ex vivo approaches. Patients don't need to have cells extracted, which can be painful and risky. They don't need to wait for cells to be modified in the laboratory, which can take weeks. They can receive treatment more quickly and easily. However, in vivo reprogramming also presents challenges. It's harder to control which cells get reprogrammed. The reprogramming might affect unintended cells. The nanoparticles need to reach the right cells and avoid being cleared by the immune system. But researchers are addressing these challenges. and in vivo reprogramming is showing promise in clinical trials. The future of cellular reprogramming might involve a combination of ex vivo and in vivo approaches. Some treatments might use ex vivo reprogramming for precision and control, while others might use in vivo reprogramming for accessibility and convenience. The choice will depend on the specific application and the trade-offs between precision and accessibility. In 2024, researchers developed AI-enhanced nanorobots that can navigate complex biological environments to detect cancer cells. These nanorobots use reinforcement learning to analyze biomarker concentration gradients and deliver drugs precisely where needed. This combines nanotechnology with artificial intelligence to create programmable medical devices at the molecular scale. Artificial intelligence is also being used to design proteins that don't exist in nature. In 2025, researchers at Evolutionary Scale and the Arc Institute used an AI model called ESM3 to design ESM-GFP, an artificial green fluorescent protein. The AI simulated 500 million years of molecular evolution to create this protein, which shares only 58% sequence similarity with its closest natural counterpart from the bubble-tip sea anemone. This shows how AI can help us program biological molecules in ways that nature never did, opening up new possibilities for medicine, environmental science, and synthetic biology. The integration of AI with nanotechnology is transforming the field. AI algorithms can process extensive datasets to predict how nanomaterials will behave, leading to faster development of new materials and applications. The global AI in nanotechnology market is projected to grow significantly, reflecting how these two cutting-edge fields are converging to create new capabilities. The possibilities are vast, but so are the challenges. Programming cells is much harder than programming computers. This might seem obvious, but the depth of this challenge is worth exploring. Computers are predictable. They do exactly what you tell them to do every single time. If you write code that says add 2 plus 2, the computer will always return 4. It won't get tired, it won't mutate, it won't evolve. It will execute the same instructions, in the same way, forever. Cells are different. They're living things. They're messy, they're noisy, they're unpredictable. They exist in a world of constant change, where molecules collide randomly, where signals fluctuate, where conditions vary. A cell is not a machine. It's a dynamic, living system. When you write computer code, you can test it, debug it, and fix it. You can run it thousands of times, and it will behave the same way every time. You can isolate bugs, trace errors, and correct mistakes. Programming is a controlled, predictable process. When you program a cell, things are different. The cell might not behave exactly as expected. It might mutate, changing its DNA. Mutations happen naturally in cells. DNA gets copied millions of times, and sometimes mistakes occur. A mutation might change your program, breaking it or altering its behavior. The cell might respond to signals you didn't anticipate. Cells are constantly sensing their environment, responding to countless signals you might not even know about. Your program might interact with these signals in unexpected ways, producing results you didn't intend. The cell might interact with its environment in unexpected ways. Cells don't exist in isolation. They communicate with other cells, exchange molecules, compete for resources. Your programmed cell might affect its neighbors, or be affected by them, in ways you didn't predict. Cells also have their own needs. They need to eat, to grow, to reproduce. They have evolved to survive, not to execute our programs. If your program interferes with the cell's ability to survive, the cell might evolve to ignore it or break it. Evolution doesn't care about your program. it cares about survival. If your program reduces survival, natural selection will favor cells that break or ignore it. This is one of the fundamental challenges of synthetic biology. How do you program cells to do what you want while still allowing them to survive and function? How do you make biological programs robust, reliable, and predictable? Scientists are working on solutions. They're designing genetic circuits that are more stable, that are less likely to mutate. They're creating systems that can detect and correct errors. They're building biological programs that can adapt to changing conditions, while still performing their intended function. They're also learning from nature. Evolution has created incredibly robust biological systems. By studying how nature solves problems, scientists can design better synthetic systems. They can borrow strategies from evolution, like redundancy, where multiple systems do the same job, so if one fails, others can take over. They can use feedback loops, where outputs regulate inputs, creating stable systems. They can implement error correction, where mistakes are detected and fixed automatically. But there's another challenge, perhaps even more fundamental. Cells are not isolated. They exist in communities, in ecosystems, in complex networks of interactions. When you program one cell, you're not just affecting that cell, you're affecting everything it interacts with. This raises important questions about safety and ethics. What happens if a programmed cell escapes into the environment? What if it interacts with natural organisms in unexpected ways? What if it evolves in ways we didn't anticipate? What if it transfers its programmed genes to other organisms? These are serious concerns, and scientists take them seriously. There are regulations governing genetic engineering. In the United States, the FDA, EPA, and USDA all have roles in regulating genetically modified organisms. There are safety protocols, containment measures, and risk assessments. Scientists are working to make synthetic biology safe, responsible, and beneficial. As of 2024, regulatory frameworks are evolving to keep pace with rapid technological advances. The FDA has established pathways for reviewing gene therapies, cell therapies, and genetically modified organisms used in medicine. The agency has approved over 20 gene and cell therapies, including CAR T-cell therapies and CRISPR-based treatments, demonstrating that regulatory systems can adapt to new technologies while maintaining safety standards. However, challenges remain. The rapid pace of development sometimes outstrips regulatory processes. New technologies like in vivo gene editing, where cells are modified inside the body, present unique regulatory challenges. The potential for off-target effects, unintended consequences, and long-term safety concerns require careful evaluation. International coordination is also important. Different countries have different regulatory approaches, creating challenges for global development and deployment of programmed cell technologies. Organizations like the World Health Organization and the International Council for Harmonization are working to harmonize regulations, but progress is slow. Ethical considerations are also evolving. As we gain more ability to program cells, questions arise about enhancement versus therapy, about equity and access, about the long-term implications of modifying biology. These are complex questions without easy answers, requiring ongoing dialogue between scientists, ethicists, policymakers, and the public. Despite these challenges, the field is moving forward responsibly. Most researchers and companies follow strict safety protocols. Containment measures prevent accidental release of programmed organisms. Risk assessments evaluate potential hazards before deployment. The track record so far has been good, with few safety incidents despite widespread use of programmed cells in medicine and industry. but the questions remain. As we gain more ability to program life, we need to think carefully about how to use that ability wisely. We need to balance the potential benefits with the potential risks. We need to ensure that our programming serves humanity and the planet, not just our immediate interests. This is an ongoing conversation, one that involves scientists, ethicists, policymakers, and the public. It's a conversation we need to have, because the ability to program cells is powerful, and with great power comes great responsibility. Let's think about specific examples of what programming cells might enable, starting with one of the most common diseases, diabetes. Imagine you have diabetes, and your body doesn't produce enough insulin. Today you might inject insulin several times a day, carefully monitoring your blood sugar, calculating doses, managing a complex routine. It works, but it's burdensome, and it's not perfect. Blood sugar still fluctuates, sometimes dangerously. But what if you could program your own cells to produce insulin on demand? What if your cells could sense your blood sugar levels and automatically produce the right amount of insulin, just like a healthy pancreas does? This would be a biological cure, not just a treatment. Scientists are working on this from multiple angles. Some researchers are developing ways to program cells in the pancreas to restore their ability to produce insulin. Others are creating new insulin-producing cells from stem cells, programming them to respond to glucose levels. Still others are programming other cell types, like liver cells or muscle cells, to produce insulin when needed. In 2021, researchers at the Salk Institute created insulin-producing cells from stem cells that could respond to glucose levels. They took stem cells, programmed them to become beta cells, the cells in the pancreas that normally produce insulin, and showed that these cells could sense glucose and produce insulin in response. When glucose levels were high, the cells produced more insulin. When glucose levels were low, they produced less. It was like having a biological feedback loop, automatically regulating blood sugar. Clinical trials are ongoing to test whether these programmed cells can effectively treat diabetes. The idea is to transplant these cells into patients, where they would function like a natural pancreas, automatically regulating blood sugar without needing injections. Other approaches involve programming cells to release insulin in response to specific signals, creating biological feedback loops that mimic how a healthy pancreas works. Scientists are developing cells that can sense glucose, process that information, and respond by producing insulin. They're creating genetic circuits that implement this feedback control, programming cells to be glucose-responsive insulin factories. The possibilities extend beyond diabetes. Scientists are developing programmed cells for other diseases, cells that produce missing enzymes for genetic disorders, cells that produce hormones for endocrine diseases, cells that produce clotting factors for hemophilia. The idea is the same. Program cells to produce what the body needs, when it needs it, automatically. This is the future of medicine, not treating symptoms, but programming the body to heal itself, not managing diseases, but curing them at the cellular level. It's medicine that works with biology, not against it, using the body's own cells as therapeutic factories. Or imagine cancer treatment. Today, chemotherapy kills cancer cells, but it also harms healthy cells. It's like using a bomb to kill a specific target. Effective, but destructive. What if you could program immune cells to recognize and kill only cancer cells, leaving healthy cells untouched? This is already happening, and it's transforming cancer treatment. CAR-T cell therapy, approved by the FDA in 2017, involves programming a patient's own immune cells. The process is remarkable. Scientists extract T cells from the patient's blood. T cells are immune cells that normally recognize and attack foreign invaders. But they don't naturally recognize cancer cells, because cancer cells are the patient's own cells that have gone wrong. So scientists program the T cells. They genetically engineer them to produce chimeric antigen receptors, or CARs. These are synthetic receptors that can recognize specific molecules on cancer cells. It's like giving the T cells new eyes that can see cancer. The programming happens in the laboratory. Scientists insert genes into the T cells that code for these CAR receptors. The cells read these genes, produce the receptors, and display them on their surface. Now the T cells can recognize cancer cells. The programmed cells are then grown in the laboratory, multiplying until there are millions of them. They're then infused back into the patient, where they seek out and destroy cancer cells. It's like sending an army of programmed assassins into the body, each one trained to recognize and kill only cancer cells. The results have been remarkable. As of 2024, 6 CAR T-cell therapies have been approved by the FDA for treating various blood cancers, with thousands of patients treated worldwide. These include therapies for B-cell acute lymphoblastic leukemia, large B-cell lymphoma, multiple myeloma, and other blood cancers. Clinical trials show remission rates of 70 to 90% for certain types of leukemia and lymphoma. Patients who had exhausted all other treatment options, who were given months to live, have gone into complete remission. Their programmed immune cells cured their cancer. The CAR T-cell therapy market has grown rapidly. As of 2024, the global CAR T-cell therapy market was valued at approximately $2.5 billion and is projected to reach $15 billion by 2030. This growth reflects both the expanding number of approved therapies and the increasing number of patients being treated. However, challenges remain. CAR T therapy currently works best for blood cancers, with limited success against solid tumors. The therapy can also cause severe side effects, including cytokine release syndrome, which requires careful management. The cost is also high, with treatments costing hundreds of thousands of dollars, though prices are expected to decrease as manufacturing processes improve and more therapies are approved. Researchers are addressing these challenges by developing next-generation CAR T-cells. These include cells with multiple CARs for targeting solid tumors, cells with safety switches for controlling side effects, and cells that can resist being turned off by cancer cells. Clinical trials are ongoing, with some showing promising results for treating solid tumors like glioblastoma and pancreatic cancer. The future of CAR T therapy also includes off-the-shelf allogeneic CAR T cells, which could be manufactured in advance and used for multiple patients, reducing costs and wait times. Several companies are developing these universal CAR T cells, with some already in clinical trials. But CAR T therapy is just the beginning. Scientists are developing next-generation CAR T cells that are more effective, that can target solid tumors, that can resist being turned off by cancer cells. They're programming cells with multiple CARs, allowing them to recognize multiple cancer markers. They're adding safety switches, allowing doctors to turn off the cells if needed. They're programming cells to produce additional therapeutic molecules, making them more powerful. The future of cancer treatment might involve armies of programmed cells, each designed for a specific patient's cancer, each optimized to maximize effectiveness and minimize side effects. It's personalized medicine at the cellular level, programming cells to fight each patient's unique disease. Or imagine aging. What if you could program cells to repair damage, to remove toxins, to maintain themselves better? Some scientists are exploring whether we can program cells to slow down or reverse aspects of aging. Aging is, at its core, the accumulation of damage in cells. Over time, DNA gets damaged, proteins misfold, mitochondria become less efficient, cells accumulate waste products. The body's repair mechanisms become less effective, and damage accumulates faster than it can be repaired. But what if we could program cells to repair this damage? What if we could program cells to remove damaged proteins, to fix DNA errors, to clear out cellular waste? What if we could program cells to maintain themselves better, to resist damage, to function optimally, even as they age? Scientists are exploring these possibilities. They're programming cells to produce enzymes that repair DNA damage. They're programming cells to clear out damaged mitochondria and produce new ones. They're programming cells to remove toxic proteins that accumulate with age. They're programming cells to maintain telomeres, the protective caps on chromosomes that shorten with age. Some approaches involve programming stem cells to be more effective at repairing tissues. Others involve programming cells throughout the body to be more resilient, to resist damage, to maintain function. Still others involve programming cells to produce factors that promote tissue repair and regeneration. The goal isn't necessarily immortality, but healthy aging. Living longer isn't the point if those extra years are filled with disease and disability. The goal is to program cells to maintain health, to function well even as we age, to extend our healthy lifespan. This is still early research, but the possibilities are fascinating. Imagine programming your cells to repair themselves, to maintain themselves, to function optimally throughout your life. It's like having a maintenance crew constantly working in every cell, fixing damage, clearing waste, keeping everything running smoothly. The implications are profound. If we can program cells to resist aging, we might be able to extend healthy human lifespan significantly. We might be able to prevent age-related diseases. We might be able to maintain physical and mental function well into old age. But this also raises questions. What would it mean to live much longer? How would society change? What are the ethical implications? These are questions we'll need to address as the science advances. But the possibility is there. Programming cells to maintain health, to resist aging, to extend our healthy years. These are still research areas, not yet ready for widespread use, but they show the potential of programming cells. If we can truly program life, we might be able to treat diseases in new ways, to extend healthy lifespans, to enhance human capabilities. But we should also think about what this means. If we can program cells, are we still working with nature, or are we creating something new? What are the limits? What should we program cells to do, and what shouldn't we? These are deep questions, and there are no easy answers. But as we explore the science, we can also explore the implications. We can think about what's possible, what's desirable, and what's responsible. The science of programming cells is still young, but it's advancing rapidly. We're like early computer programmers in the 1950s, just beginning to understand what's possible. The computers of that era were huge, slow, and limited. They filled entire rooms, consumed massive amounts of power, and could perform only basic calculations. Programming them required deep expertise, and the programs were simple. Today's computers are tiny, fast, and powerful. They fit in our pockets, consume minimal power, and can perform billions of calculations per second. Programming them is accessible to millions of people, and the programs can be incredibly complex. The transformation has been remarkable. We can only imagine what programming cells might look like in 50 years. But we can see the direction. The tools are getting better, the success rates are increasing. The applications are expanding. We're moving from simple programs to complex systems, from single cells to multicellular communities, from basic functions to sophisticated behaviors. Imagine a future where programming cells is as common as programming computers, where you can download genetic code from a repository, modify it for your needs, and upload it into cells, where biological programs are shared, improved, and combined, just like software today, where you can program cells using familiar programming concepts without needing deep expertise in molecular biology. Imagine a future where cells and computers work together seamlessly, where biological sensors feed data to computers, where computers process information and send instructions back to cells, where the boundary between biology and technology becomes blurred, where living and non-living systems integrate into hybrid systems with capabilities beyond either alone. Imagine a future where we can program our own bodies, where we can enhance our cells, where we can program them to fight disease, to repair damage, to maintain health, where aging becomes optional, where disease becomes rare, where our biological capabilities are limited only by our imagination. These futures might seem distant, but the foundations are being laid today. Every experiment, every breakthrough, every new tool brings us closer to these possibilities. The science is advancing, the tools are improving, and the future is coming faster than we might expect. But we're already seeing remarkable progress. In 2024, researchers at IIT Indore developed quantum AI nanotechnology systems that combine quantum transport with artificial intelligence to detect genetic mutations more accurately. These systems use solid-state nanopores, tiny holes just nanometers wide, to measure electric signals as DNA strands pass through. As each DNA base passes through the pore, it creates a characteristic electrical signature. AI algorithms analyze these signatures to identify the sequence and detect mutations with unprecedented accuracy. This allows for earlier and more precise diagnostics, showing how nanotechnology can enhance our ability to read and understand genetic information. The system can detect single base pair mutations, identify genetic variants, and diagnose diseases earlier than ever before. It's like having a molecular-scale DNA reader, powered by AI, that can analyze genetic information with incredible precision. Scientists have also created luminescent nanocrystals that can rapidly switch between light and dark states. These nanocrystals can change from emitting light to being dark in nanoseconds, and back again, billions of times per second. This rapid switching enables new types of optical computing, where information is encoded in light rather than electricity. These nanocrystals pave the way for faster and more efficient data processing, with potential applications in high-density optical computing and AI-driven data centers. Optical computers could process information at the speed of light, using far less energy than electronic computers. This shows how nanotechnology isn't just about biology, but also about creating new types of computers that work at the molecular scale. In March 2024, researchers published work on transfer learning approaches to control cell behavior. Transfer learning is a technique where AI models trained on large datasets are adapted for specific tasks. By pre-training AI models on transcriptomic data from thousands of cells, scientists can tailor models to specific reprogramming goals. This means AI can help predict how to program cells to become specific cell types, making cellular reprogramming more reliable and efficient. Instead of trial and error, scientists can use AI to predict which genetic programs will work, which will fail, and how to optimize them. It's like having an AI assistant that understands cellular programming, that can help design genetic circuits, that can predict outcomes before experiments are run. The integration of AI with cellular programming is accelerating progress. I can design genetic circuits, predict their behavior, optimize their performance. It can analyze vast amounts of biological data to discover new programming strategies. It can simulate cellular behavior, testing programs in silico before trying them in cells. The combination of AI and cellular programming is creating new capabilities, enabling progress that wouldn't be possible with either alone. Advanced microscopy techniques now allow scientists to see structures smaller than 1 nanometer. Scanning electron microscopy, transmission electron microscopy, and atomic force microscopy have achieved resolutions below 1 nanometer. These tools are essential for analyzing how programmed cells and nanomaterials actually work, allowing scientists to verify that their genetic programs are executing correctly at the molecular level. Scanning electron microscopy uses a beam of electrons to create images of surfaces, achieving resolutions down to about 0.5 nanometers. Transmission electron microscopy passes electrons through samples, achieving even higher resolutions, down to about 0.1 nanometers. Atomic force microscopy uses a tiny probe to feel surfaces, achieving resolutions down to atomic scales. These techniques allow scientists to see individual molecules, to observe how DNA folds, to watch how proteins interact, to verify that genetic programs are working as designed. They can see how nanoparticles interact with cells, how DNA origami structures assemble, how programmed cells change in response to signals. This level of observation is crucial for understanding and improving cellular programming. But seeing isn't enough. Scientists also need to understand how things work, not just what they look like. This requires techniques that can observe processes in real time, that can measure forces, that can detect chemical changes. Scientists are developing new techniques that combine imaging with other measurements, creating comprehensive views of how programmed cells function. For example, scientists can now observe cells in real time using fluorescence microscopy, watching as genetic programs execute, as proteins are produced, as cells respond to signals. They can measure forces using atomic force microscopy, understanding how cells interact with their environment. They can detect chemical changes using spectroscopy, monitoring metabolic processes as genetic programs run. These observational techniques are essential for debugging genetic programs, for understanding why they work or fail, for improving their performance. Just as computer programmers use debugging tools to understand their code, cellular programmers use microscopy and other techniques to understand their genetic programs. The ability to observe and measure is crucial for advancing the field. The field is also moving toward more sustainable approaches. Green nanotechnology focuses on creating biodegradable nanomaterials from renewable sources. This is important because, as nanotechnology becomes more widespread, we need to ensure it doesn't create new environmental problems. Startups like Nanomatics in Singapore are developing recycled carbon nanotubes from plastic waste, reducing environmental impact while creating useful materials. These recycled nanotubes can be used in energy storage and electronics, reducing the need for virgin materials and cutting down fossil resource consumption. The process involves breaking down plastic waste and reforming it into carbon nanotubes, creating valuable materials from waste while reducing environmental impact. Scientists are using plant-based materials to synthesize nanoparticles, moving away from toxic chemicals. Traditional nanoparticle synthesis often uses toxic chemicals and high temperatures, creating environmental hazards. But researchers are developing green synthesis methods that use plant extracts, bacteria or fungi to produce nanoparticles. These methods are more sustainable and produce less waste. For example, researchers have developed smart barrier nanocoatings for food packaging that provide plastic-like barrier properties to compostable materials, increasing shelf life without altering biodegradability. These nanocoatings are made from natural materials and can be composted along with the packaging, reducing plastic waste, while maintaining the protective properties that make plastic packaging useful. The move towards sustainability is driven by both environmental concerns and economic considerations. Sustainable materials can be cheaper to produce, especially as regulations on waste and emissions become stricter. They can also create new markets, as consumers increasingly prefer sustainable products. Companies that develop sustainable nanotechnology solutions are finding competitive advantages in the marketplace. This shows that as we learn to program matter at the smallest scales, we're also learning to do it responsibly. The field is maturing, moving beyond just what's possible to what's sustainable, what's responsible, what's beneficial for both people and the planet. This is a positive trend, and it's essential for the long-term success of nanotechnology and synthetic biology. The convergence of synthetic biology and nanotechnology is creating new possibilities. By combining programmable cells with nanoscale delivery systems, scientists can create more precise and effective therapies. For instance, researchers are developing nanoparticles that can sense disease markers and trigger programmed cells to produce therapeutic molecules only when needed. This creates a feedback loop where the treatment responds dynamically to the patient's condition, potentially reducing side effects and improving outcomes. This convergence is being formalized in a field called synbio nanodesign, which integrates synthetic biology and nanotechnology for next-generation targeted drug delivery systems. These systems utilize engineered nanomaterials to create specialized drug carriers capable of dynamic, stimuli-responsive, and site-specific therapeutic delivery. Research published in 2024 shows that these hybrid systems can improve drug efficacy by 50 to 80%, while reducing side effects by similar amounts. The integration is also creating new diagnostic capabilities. Scientists are developing doctor-in-a-cell concepts, where autonomous, programmable molecular devices operate within the human body to perform diagnostic and therapeutic functions. These molecular computing devices can analyze their environment and release appropriate treatments, potentially replacing traditional drugs with smart drugs that respond to the body's needs in real time. DNA walkers, nucleic acid nanomachines capable of moving along tracks and performing tasks like cargo transport, are being developed for applications in diagnostics, targeted therapy, and biosensing. These nanorobots can navigate through the body, detect disease markers, and deliver therapeutic payloads precisely where needed. The convergence of AI, synthetic biology, and nanotechnology is accelerating progress. AI-driven tools facilitate automated design, build, test, and learning cycles for engineered biological systems. This is democratizing synthetic biology, making it more accessible to researchers without deep expertise in molecular biology. However, this convergence also presents challenges related to reliability, dual-use concerns, and governance, necessitating updated regulatory frameworks and responsible development practices. But we can see the direction. Scientists are getting better at reading DNA, at writing DNA, at editing DNA. The cost of DNA sequencing has dropped by a factor of 1 million since the Human Genome Project began. The cost of DNA synthesis is also dropping rapidly, making it cheaper to write genetic code. The precision of gene editing is improving, with new CRISPR variants that are more accurate and have fewer off-target effects. They're getting better at understanding how cells process information, at designing biological circuits, at programming cell behavior. Our understanding of biological circuits is deepening, with researchers mapping out how genes interact, how proteins regulate each other, how signals flow through cells. This understanding is enabling better circuit design, more reliable programming, more sophisticated behaviors. The tools are improving, the knowledge is growing, and the possibilities are expanding. New tools are being developed constantly, new techniques are being discovered. New applications are being explored. The field is advancing rapidly, and it's exciting to watch. But progress isn't always linear. There are setbacks, failures, unexpected challenges. Some genetic programs don't work as expected. Some applications face regulatory hurdles. Some technologies prove more difficult than anticipated. But overall, the trend is positive. We're getting better at programming cells, and the applications are expanding. The convergence of different technologies is also accelerating progress. AI is helping design genetic circuits. Nanotechnology is enabling better delivery of genetic programs. Advanced microscopy is allowing us to observe and understand what's happening. The combination of these technologies is creating new capabilities that wouldn't be possible with any one technology alone. This convergence is creating a positive feedback loop. Better tools enable better understanding, which enables better tools, which enables better understanding. Each advance builds on previous advances, accelerating progress. We're in an exciting time, where rapid progress is possible, where new applications are emerging constantly, where the future is being written right now. Some researchers imagine a future where programming cells is as common as programming computers, where you can download genetic code from repositories like GitHub, modify it for your needs, and upload it into cells, where biological programs are shared, improved, and combined, just like software today, where there are libraries of genetic code for common functions, where you can build complex programs by combining simpler ones, where programming cells is accessible to anyone with basic biology knowledge. This future would democratize cellular programming, making it accessible to researchers, entrepreneurs, and even hobbyists. Just as personal computers democratized computing, making it accessible to millions of people, programmable cells could democratize biology, making it possible for more people to create biological solutions to problems they care about. Others imagine a future where cells and computers work together seamlessly, where biological sensors feed data to computers, where computers process information and send instructions back to cells, where the boundary between biology and technology becomes blurred, where living and non-living systems integrate into hybrid systems with capabilities beyond either alone. This convergence could create new types of systems. Imagine a computer that uses biological components for certain tasks, like pattern recognition or energy production. Imagine biological systems that use computer components for certain tasks, like data storage or computation. The combination could be more powerful than either alone. Still others imagine a future where we can program our own bodies, where we can enhance ourselves, where we can become something more than purely natural. This raises profound questions about what it means to be human, about the relationship between nature and technology, about the future of life itself. If we can program ourselves to be stronger, faster, smarter, healthier, what does that mean for human nature? If we can enhance our capabilities beyond what evolution provided, are we still human? These are deep questions that don't have easy answers. But they're questions we'll need to grapple with as the technology advances. These are big ideas, and we're just beginning to explore them. But the science is real, the progress is happening, and the future is coming. The ability to program cells is one of the most powerful technologies we're developing, and it's going to change everything. How it changes things, and whether those changes are positive depends on how we use this power, on the choices we make, on the values we prioritize. The future isn't predetermined. We're writing it right now, through our research, through our applications, through our choices. The ability to program cells gives us new power, but with that power comes responsibility. We need to use it wisely, to benefit humanity and the planet, to create a future that's better than the present. As we drift toward the end of this exploration, let's return to the simple question we started with. What if we could program cells like we program computers? The answer is that we're already starting to. Scientists are programming cells to produce medicines, to detect diseases, to clean up pollution, to build materials. They're creating biological circuits, genetic programs, and synthetic biological systems. They're using nanotechnology to deliver instructions to cells, to program molecules, to create materials with amazing properties. Every day, in laboratories around the world, scientists are writing genetic code, designing biological circuits, programming cells to do new things. They're treating diseases, cleaning environments, creating materials, all by programming life itself. But we're still at the beginning. Programming cells is harder than programming computers. Cells are living things, with their own needs and behaviors. They're part of complex ecosystems, with interactions we don't fully understand. They evolve, they mutate, they surprise us. Yet the potential is enormous. If we can truly program life, we might be able to solve problems that seem impossible today. We might be able to treat diseases in new ways, to clean the environment, to create new materials, to enhance human capabilities. We might be able to program our own cells to fight cancer, to produce insulin on demand, to repair damage, to slow aging. Imagine a world where diseases are cured by programming cells, not by drugs. Where pollution is cleaned by programmed bacteria, not by mechanical processes. Where materials are grown by programmed cells, not manufactured in factories. Where our bodies maintain themselves, repair themselves, optimize themselves, all through programmed cells. The science is fascinating, the possibilities are exciting, and the questions are profound. As we learn more about how to program cells, we're also learning more about how life works, about how information flows through biological systems, about how simple rules can create very complex behavior. We're learning that cells are already biological computers, processing information, following instructions, and responding to their environment. We're learning that we can write new instructions, create new behaviors, design new biological systems. We're learning that the boundary between biology and technology is not as fixed as we once thought. We're discovering that programming cells is not just about controlling biology, but about understanding it. Every time we program a cell, we learn something new about how cells work. Every genetic circuit we design teaches us about biological circuits. Every programmed cell reveals something about the nature of life itself. The future of programming cells is still being written. But the story has begun. And it's a story worth following. It's a story about understanding life, about controlling biological systems, about creating new possibilities. It's a story about the convergence of biology and technology, about the future of medicine, industry, and our relationship with the natural world. It's also a story about responsibility. As we gain the ability to program life, we must use that ability wisely. We must consider the implications, the risks, the ethics. We must ensure that our programming serves humanity and the planet, not just our immediate interests. But the potential is too great to ignore. The ability to program cells could transform medicine, could solve environmental problems, could create new materials, could enhance human capabilities. It could help us understand life itself, could help us work with nature rather than against it, could help us create a better future. So the next time you think about a cell, remember that it's already a tiny biological computer, processing information, following instructions, and responding to its environment. And remember that scientists are learning to program these computers, to write new instructions, to create new behaviors. The future is coming, and it's going to be fascinating. Cells and computers, biology and technology, nature and design, all converging in ways we're just beginning to imagine. The ability to program life is one of the most powerful technologies we're developing, and it's going to change everything. But for now, as we drift towards sleep, let's just appreciate the wonder of it all. The fact that we can program cells, that we can write genetic code, that we can control biological systems. The fact that biology and computing are converging, that we're learning to program life itself. It's remarkable, it's exciting, and it's just beginning. The future of cellular programming is being written right now in laboratories around the world by scientists who are pushing the boundaries of what's possible. And as we sleep, as we dream, those scientists are working, discovering, creating, programming cells to do things we can only imagine. Sweet dreams, and keep wondering about the amazing possibilities that science brings us. The ability to program cells is transforming our world, and the best is yet to come. Sweet dreams, and keep wondering about the amazing possibilities that science brings us.